Right. Today we are going to talk about antibody. What is antibody? Antibody is a plasma protein which are produced by the B lymphocyte. What is antibody? Antibody is a plasma protein which is produced by the B lymphocyte. As we know that this is blood vessel. This is my blood vessel. This blood vessel contain blood. What is blood? What is the composition of blood? Blood is composed of two parts. One is called blood cell and the other is called blood plasma. Blood cell are white blood cell, red blood cell, and platelets, while the remaining part in the blood is called blood plasma. Blood plasma consists of water, vitamin, mineral, waste product, dissolved oxygen, dissolved carbon dioxide, and different types of protein. So, what is plasma protein? The protein which is present and the blood plasma is called blood plasma protein. There are different types of blood plasma protein. There are mainly three types of blood plasma protein. These proteins are albumin, fibrinogen, and globulin. These are the three main types of protein which is called plasma protein. These are the three main types of plasma protein: albumin, fibrinogen, and globulin. Okay, these are the three types of plasma protein. Then globulin have several types. Gamma globulin. Globulin is the main plasma protein, which form about 75% of the plasma protein. This is the main plasma protein, which consists of in large amount, which is present in the blood in large amount, which make about 75% of blood volume of blood plasma protein. Okay, globulin have different types: gamma globulin, beta globulin, alpha 2 globulin, alpha 1 globulin. These are the types of globulin protein. But this beta globulin also have many types. This also have many types. Alpha 1 also have many types. But gamma globulin, what is gamma globulin? Gamma globulin, this is the actual antibody. What is gamma globulin? Gamma globulin is called the antibody. Okay, gamma globulin, this protein is called antibody. So these are the also globulin protein. This is also the globulin protein, but this gamma globulin protein is called antibody. So what is antibody? Antibody is a plasma protein, is a globulin protein. These are all the globulin protein, but this is the gamma globulin protein. So antibody is called gamma globulin protein. Antibody is also called immunoglobulin, or antibody also called gamma globulin, because this is the globulin protein, but the gamma globulin protein. Okay, why it is called immunoglobulin? Immunoglobulin is a functional term because this is the protein which help in the protection of the body. This is the protein of the immunity system. Therefore, it is called immunoglobulin. This is called immunoglobulin because this is the part of the immunity system. This is the functional term, while the gamma globulin is a physical term. Immunoglobulin is a functional term, while the gamma globulin is a physical term because this antibody is physically made from the gamma globulin. So gamma globulin antibody is also called gamma globulin and also called immunoglobulin. This is a functional term. This is a physical term. Okay. This is the gamma globulin. Gamma globulin. This all of them have different types, but the gamma globulin are also phenotypes. Antibody IgD, IgE, IgM, IgA, IgG. These are the period classes of antibody. I am not going on detail for this because for the classification, we will classify antibody and detail in the next class. In the next class, I will in the part 2. This is the part 1. In the part 2, I will explain antibody classification and its function in detail. But okay. What is plasma protein produced? Antibody is clear. Antibody is a plasma protein which is produced by the B lymphocyte. Antibody plasma protein derivation. Where is plasma protein derived? Where is plasma protein as produced? Plasma protein derived from liver mainly. Plasma protein mainly produced by the liver, bone marrow, and spleen. But the gamma globulin is a plasma protein which is produced by the B lymphocyte. These all protein are produced by the liver, spleen, and bone marrow. But the gamma globulin is a protein is a plasma protein which is only produced by the B lymphocyte during the infection. It is also present in the normal condition before the infection, but it is mainly produced by the B lymphocyte during the disease condition. So this protein is produced by the B lymphocyte. How it is produced by the B lymphocyte? How this protein is produced by the B lymphocyte? This is a bacteria. This is a bacteria which have many antigen on its own surface. Different types of antigen: triangular, rectangular, round antigen. This is a bacteria. When this bacteria attack our body, then macrophage in our body. This is a macrophage. Macrophage engulf this bacteria. This is bacteria. This is a macrophage. Macrophage engulf this bacteria and destroy them. This bacteria. Then this macrophage express this antigen on its own cell membrane. Expose this antigen on this bacteria is engulfed and destroyed out into different parts, but the antigen of the bacteria is exposed on its own membrane, and these antigen are presented to the antigen helper cell, antigen presenting cell. Sorry, this is the antigen presenting cell. This antigen is presented to the T helper cell. T helper cell. T helper cell. When this antigen is presented to the T helper cell, this is the T helper cell. T helper cell recognize this antigen with the help of its own receptor is called TCR. TCR is a receptor which is present on the T helper cell. This is the TCR. When this macrophage present this antigen to the T helper cell, then the T helper cell recognize this antigen and this antigen when the T helper cell recognize this antigen, and then after a long process, antibody is produced. Antibody is produced, and this is the antibody which is produced against this bacteria. By this way, antibody is produced by the B lymphocyte, and this antibody then destroy. There are present million of bacteria. Then this antibody destroy attached to this bacteria and destroy this bacteria. So antibody are produced in this way. Okay. Shortly, T helper cell activate B lymphocyte. This is the B lymphocyte. B lymphocyte, when activated, this B lymphocyte is divided into two cells: B plasma cell and B memory cell. B plasma cell and B memory cell. Then this B plasma cell produced antibody. T helper cell stimulate B plasma. Antigen stimulate T helper cell. T helper cell stimulate B cell, B lymphocyte. Then B lymphocyte, this is the B lymphocyte. B lymphocyte is divided into two cells: B memory cell and B plasma cell. B plasma cell produce antibody. This is the actual production of antibody. B plasma cell produce antibody, and the antibody fight against this bacteria. Okay, this is the production of antibody. Now, structure of antibody. What is the structure of antibody? Structure of antibody. Antibody consists of structure. This is the very simple structure. This is a very simple model. Antibody consists of four chain. This is one chain. This is another chain. This is another chain. This is another chain. So antibody mainly consists of four chain. Two light chain and two are heavy chain. Antibody consists of two light chain and two heavy chain. And these two heavy chain is held together by disulfide bond, and heavy chain and the light chain are held together by the disulfide bond. This is the disulfide bond, okay, which combine these chain with each other. This is the four chain: two heavy chain, two light chain. But antibody also consists of two region: constant region and variable region. What is constant region? What is variable region? This is the variable region. Variable region is composed of half. This is the light chain. Half this region is called this region. This is called the variable region. This part and this part, half of the light chain, and this is the heavy chain. 1/4 of heavy chain and 1/2 of light chain make the variable region. This is when we divide this into four parts. This is part 1, part 2, part 3, part 4. When we divide this into four parts. Part 1, okay. Part 1, part 2, part 3, part 4. 1/4. 1/4 plus 1/2. 1/2 of the light chain and 1/4 of the heavy chain. At what terminal? At the amino terminal. This is the amino terminal. Variable chain is made from the light 1/2 of light chain and 1 pore of heavy chain at the amino terminal. What is mean by amino terminal? Heavy chain, light. This is a light chain. This is the light chain. This is the simple structure. This is the light chain. This is the heavy chain. Light chain consists of about 1 to 220 amino acid. This chain is made from about 220 amino acid, 220 amino acid, while this chain is made from 1 to 4 5 amino acid. Okay, light chain is made from 220 amino acid, and the heavy chain is made from 450 amino acid, 450 amino acid. So this is the amino acid number 1, this is amino acid number 1, this is amino acid number 2, and so on, 20 to 0. Okay, so the light chain, this is the this is the variable region. This is the variable region. Variable region consists of the amino terminal of carbon of the amino acid number 1 of the light chain, and amino terminal of the amino acid number 1 of the heavy chain. Okay, what is mean by the heavy by the variable region? Variable region consists of the 1 by 2 and 1 by 4 of these two chain of the amino terminal. Okay, but this is called this is the heavy region. This is the variable region. Why it is called variable region? Because the arrangement of amino acid, the placement of amino acid, and the number and the sequence of amino acid in this region is variable. Because different types of variable region. One types of because they are attached to different types of antigen. This is also called antigen binding site. This is the antigen binding region, because the antigen is bound to this site. When antibody attack on the antigen, when the antibody attack on the microbe, when the antibody attack on the bacteria, etc., then these two parts is attached to the bacteria, antigen, etc. So this is called the antigen binding site, pathogen binding site, pathogen binding region, antigen binding region, variable region, because the placement, the arrangement, and the number of amino acid in this region is variable. Okay, this is a bacteria. Here, the antibody is formed from the previous concept. Antibody is formed. This is a bacteria. Then the antibody come and attach to this bacteria. Suppose this is a bacteria. I enlarge this. This is a bacteria. This is a bacteria which have pathogen, which have antigen 1 and antigen 2. This is a bacteria which have this antigen and is attacked by the antibody. Then okay, this bacteria, this is the variable region. This is the variable region. Okay, this is the variable region, and this is the constant region. When it is attached to this bacteria or antigen, then this antibody is attached to with the help of variable region. Okay, this is the with the help of variable region. This is a constant region. This is the variable region which attached to this antigen. Okay, so this is called antigen binding region. But this region is called the constant region. What is mean by constant region? Constant, and this region, the number of amino acid, the sequence, the placement of amino acid, and all the antibody is same. But and different antibody, this area have different types of amino acid. But the number is constant always. But there the placement, arrangement, sequence is different in different antibody. But in all antibody, this region have same sequence, same number, same placement of amino acid. Therefore, this is called so therefore this region is called constant region. Okay, this is the variable region. This is the constant region. Constant region consists of 1 2 by of a light chain and 3 up to 3 by 2, 1 up 2 of light chain plus 3 by 4 of heavy chain make constant region. It is also called FC portion. What is mean by FC portion? It is also called this region. What is also called it is also called FC region. Why it is also called FC region? FC mean F mean fragment, C mean crystallizable. This is the crystallizable part. FC mean it is also called FC constant region is also called FC region. Why it is called FC region? Because FC means fragment crystallizable region. Why it is called crystallizable region? Because the antibody have a specific region which is called hinge region. This is the antibody which have a specific region which is called hinge region. What is hinge region? At the hinge region, antibody is broken down by the addition of peptide enzyme. Suppose this is a container which have antibody. When we add peptide, P-A-P-I-A and peptide enzyme. When we add peptide enzyme to this container which consists of the antibody, then and also add antigen. When we add peptide enzyme and antigen, then this is antibody. This is a antibody which is held together with each other by disulfide bond. Then this antibody is broke down into two portions, three portions. One portion is here, other portion is here, and the other portion is here. 1, 2, and 3. This antibody is broke down into four or three portions. 1, 2, 3. This is called. This is the region. This is the heavy region portion. Sorry, this is the variable region portion. Variable. So this peptide enzyme break down this bacteria and this antibody at this point. So it is converted into three region. This is the region. This is the region, and this is this region. Okay. So the antigen is attached to this region. Antigen is attached to this region. Antigen is attached to this variable region. This is the antigen binding site. While this region is not attached to the antigen. This is a constant region. These region of main antibody settle down and become crystal and make here a crystal. So this region and this region attached to the antigen, but this become a crystal, and this region pre-fluid and the fluid present in this container. So this region is also called FC portion, fragment crystallizable portion. Okay. This region is called constant region. This region is called FC portion, FC region. This region is also called complementary fixation region. Complementary fixation region. It is also called complementary fixation region because this region start complementary system. I am not going on detail for the complementary system because I have already uploaded a detailed lecture on the complementary system, but just giving a simple idea. This is a bacteria. This is a bacteria which have this one is a one antigen and this one is another antigen. This is a bacteria. Okay, this is a bacteria. When antibody is attached, this is antibody. When antibody is attached to this antigen, this is the antigen. This is bacteria. This is the variable region. This is the antigen. This and these are the antigen, while this is the constant region. This is the constant region. How it start complementary system? Complementary fixation. This is also called complementary fixation region because when this in the complementary system, when this antibody is attached to the bacteria, then there are about 30 different types of complementary protein. Then complementary protein number 1 come and attached. Complementary protein number 1 come and attached here by a specific loop. When this antibody is attached here, then there come a conformational changes and produce a specific loop of constant region where complementary protein number 1 is attached and so on. This all protein is make a ring which is called complementary pathway. This start a complementary system. So complementary system is start from this point, from constant region. When antibody attached to the bacteria, they produce a specific site. This specific site complementary protein are attached by a series of reactions, and the complementary system is Let's start. This is the region of constant amino acid. This is the region which start complementary system, but this is the region which through which antibody is attached to the cell surface. Antibody is attached to the bacteria with the help of to the antigen to the other microbe with the help of this region. This is a bacteria now. This is antibody. This antibody is attached to the bacteria with the help of variable region. But in our body, some types of cell like mast cell have antibody on its own surface. Some people also have this concept that the B lymphocyte have a receptor called B cell receptor BCR. Some people call this this BCR is a antibody. But and mast cell have a specific antibody. I am not going a detail for the mast cell because the mast cell is already explained in very detail and big detail in the allergy topic. I have already uploaded this topic, okay? But the mast cell, this is the mast cell. This is the allergic cell which are having a specific antibody on its own surface which produce allergic response. This is the antibody which is present on the mast cell and attached to a specific receptor in the body. So in the slightly abnormal condition, I am not going in detail, but mast cell have a specific antibody and the which is attached by this portion. This is a mast cell which have antibody which is attached to with the help of constant region. So this is the antigen binding site. This is the cell binding site. This is the constant region. This is the Fc portion. This is the fragment. This is the complementary system fixation part. So these are all about the structure and production of antibody in the part 1. In the part 2, I will discuss you the classification of the antibody and the function of different antibody, the function of this and this. I will explain you in the next lecture.